All right. Welcome, everybody. Welcome to the Polar Steam live stream from the Nathaniel B. Palmer. We're super excited to have everybody here today. While we are waiting, we'll give just a couple more seconds here for folks to join. While we're waiting, please post in the chat where you're joining from and how many folks are in your location. We'd love to see where people are joining from. Post in there and we'll give it just another minute or two. Welcome everybody. Excited to have you on board. Can see we've got our fellows on the Palmer and we'll introduce them in just a minute here. All right. Well, I'm Melissa Barker. I'm the Outreach and Engagement Lead for Polar Steam for the Polar Steam program. And I'm excited to have everybody here with us today. Polar Steam is an NSF-funded program based at Oregon State University, and we facilitate virtual and field-based collaborations between polar researchers, educators, and artists and writers to enhance science communication and broaden the reach of polar science. A recording of today's live stream will be posted on our YouTube channel. We will keep the focus of today's event on the science and the experience on the ship. If you have questions about our artists and educator program, our artist program, our educator program, our researcher programs, please refer to the informational webinars on this channel and on our website. We are really excited to have our folks on the Palmer with us today. So again, if you are joining us, please tell us where you're joining from and how many people are in your location. All right, so we've talked about Polar Steam. Okay. Today we're excited to connect with our three Polar Steam Fellows aboard the Nathaniel B. Palmer in the Southern Ocean. We'll start with a presentation from the fellows and follow that with a question and answer session. We invite folks to submit questions in the live chat. Our team will monitor these questions and then we'll address them and any questions that were submitted ahead of time through the Google form after their presentation. Again, we want to keep those questions focused on their experience on the ship and the science that they're doing. So I'd love to introduce Educator Fellow Amanda McBride, STEM teacher at Linus Pauling Middle School in Corvallis, Oregon. Educator Fellow Kylie Preheim, a 7th grade science teacher at Chisholm Middle School in Newton, Kansas. And Researcher Fellow Becky Robinson, Becky Robinson, Professor of Oceanography from the University of Rhode Island. So now I'll turn it over to you guys. And I will mention they are live from the Palmer, so we are getting a little bit of internet delay here and there. I see that they are frozen at this moment, but hopefully they will be able to. I'm gonna see if getting rid of my video might be helpful. All right, you guys are on.
Okay, thank you. All right.
Good morning and good afternoon,
yes.
Everyone. Thanks for joining.
Hello. We have a little bit of information about each of us here. We're not going to take time.
Thanks for bearing with us, folks. We are experiencing a little trickiness with the internet, so yeah, hopefully it will come back here in a second. Are you guys back with us?
All right.
Okay.
Yeah.
Yep,
Okay.
go for it.
All right. I'm Amanda McBride. I am the STEM teacher at Linus Pauling Middle School. And I am a Polar Steam Research Education Fellow, coming from Corvallis, Oregon, but currently on the Natty B.
I'm Kylie Preha.
Thanks again. Oh, you're back.
We're going to tell you a little bit about what we've seen and what we've done out here while we've been at sea for the last 6 weeks. Change slides. So the reason we came out here to the Southern Ocean is to
Thanks again for your patience, folks. Oh, okay, you guys are back again.
So one of the things that we need for looking back in climate are really good tools. The Southern Ocean is super important for the climate system, and the sediments underneath the Southern Ocean that we would use for record. So and so we're sampling the modern water column, modern diatoms, modern plankton to try to understand better how the surface ocean characteristics can be recorded by these organisms called diatoms. So they build their shells out of materials and matter from the surface ocean, such as silicon and nitrogen and other elements, and their chemistry reflects these changes. So the picture over to the right on this slide is a sort of a hypothetical of what the Southern Ocean, what the Southern Ocean's role was in the past. So this is in the last ice age. We think that the Southern Ocean had maybe less nutrients and lots and lots of dust, and this made it so that less CO2 sunk out of, or less CO2 was in the atmosphere. More CO2 was captured by the ocean. And we want to understand if we can improve upon this picture. Can we make better inferences about whether the circulation in the ocean or the ocean biology are playing a bigger role in drawing down atmospheric CO2? Next slide. So I said diatoms were super important in the Southern Ocean. They're the dominant primary producer in the Southern Ocean, and this is a huge ecosystem, and they're the base of this food web. Globally, diatoms are they're cosmopolitan, and globally they play a really important role in carbon and oxygen cycles. So because they photosynthesize, they produce oxygen, and it's estimated that they are responsible for about every 5th breath that you take. And so we are we're studying diatoms in particular because of their importance in the sediment, but also their importance in the global carbon cycle. You can see in the middle here some pictures that we've taken on the ship under the microscope. The first one on the top is an organism, a plankton called a dinoflagellate. And inside, you can see that it was eating diatoms. And so for scale, perhaps the diatom inside of this dinoflagellate is shown in this lower microscope image down here, something similar at least, not the same one. And we have big diatoms and tiny diatoms that we're studying in the ocean. Next slide. So to do this work, we have actually crossed almost the entire Pacific Basin. We started out down here at the very tip of Chile in Punta Arenas. And we went out the Straits of Magellan, crossed the dreaded Drake Passage in the roaring sort of 40s and 50s. We were held up, in fact, because of a nice giant storm. And we made our way to Palmer Station. This is one of the US bases on Antarctica. And we were lucky enough to get to spend a day or two on Palmer Station to drop off some people and exchange some cargo. And then we headed out across the Pacific. Each one of these points here is a point where we actually did what we were calling a bucket sample. I mean, we literally threw a bucket over the side of the ship because we wanted to sample the slush and the sea ice. And so this map is actually a temperature map. And you can see in the white here, it's all below freezing. So we were traveling along the ice edge because it's really stable. The ice helps to dampen the swell, and it's a comfortable ride. It's also great for checking out penguins and seals and sort of looking at really interesting scenery. Finally, we made it across to our transect, which is where we worked. So that's where all these little dots are. So these diamonds are, each one of these is one of our stations. There's one every degree of latitude along 170 West. And that's where we started to do like really the bulk of our science and to sample the ocean. And so next slide. So we have been sampling the ocean to try to learn how well diatoms record the chemistry, as I said. And the first steps of that are happening on the ship. And so one of the things that we're doing is sampling the water column and looking at who's there, what diatoms are in the water. So over here on the left, I'm showing sort of the relative abundance of different diatom species versus latitude. So we took a stop at every degree of latitude. Somebody took a sample and they counted diatoms on a slide. And you can see that down here at the furthest southern points, we're dominated by sea ice diatoms. And then actually, interestingly, it goes down and it comes back up again, which is, there was sea ice here in the past like this season, but maybe not, but maybe not, we think of this as not being a sea ice dominated place. And you can see here that the sea ice diatoms are lower as we get north, and the open ocean and other diatoms are taking over. Similarly, as we go across the Southern Ocean from south to north, we can see real changes in the amount of nutrients that are in the surface ocean. And both in terms of nitrogen, so nitrate, and silicon, two nutrients that are absolutely essential to diatoms' growth. they go from high to low in the surface ocean. And you can see from these red colors that there's lots and lots of nutrients in the deep ocean. If you think about photosynthesis, organisms use carbon, nitrogen, phosphate, in the case of diatoms, silicate. And so where there's high nitrogen and silicon, it probably means there's high carbon. And so so this is one way you can think of how the Southern Ocean is important to the carbon cycle. This high nutrient water is almost touching the surface. And so this is how CO2 escapes to the atmosphere. Next slide.
Okay, so I'll take over here. This is Kylie. We have some photos here of the journey that we've been on. As Becky described, we've kind of covered the whole Pacific Basin down here, but we have a photo starting on the left here. This is a photo taken of Amanda in front of our ship whenever we were still in port at Punta Arenas. And then kind of the little inset photo there I have of me getting my little chlorophyll filtering station set up. So when we were in Punta Arenas, we were unpacking all of our science boxes and getting everything set up. And then once we set off, we crossed the Drake Passage. So there's a photo of myself and two of the awesome folks that I'm working with crossing the Drake Passage. And then we made a stop in Palmer Station, Antarctica, which is one of the 3 US Antarctic bases. And we stopped there because we had some folks on board that were going to be working at Palmer Station. So they got off there and we actually got the chance to go and tour the station and walk around a little bit. So that photo there in the middle is of most of the science team. I think we're missing a couple people in front of the Palmer Station sign. And then after we left Palmer Station, we were there for about 3 days. We started to transit across the Southern Ocean to our research transect, which, as Becky mentioned, is 170 West. So that picture there, which says transiting to 170 West, that's a picture taken as we were kind of in the sea ice, and it was a good photo opportunity. But during this time, we were doing a lot of preparation. We were hearing some science talks from folks, and we were really getting ready to start our science. We were also working on a program called Visathon, which we'll talk more about in a later slide here. But we were thinking about the ways that we as scientists and teachers can also work with artists to represent our data and our science visually. This last picture here is, well, it's actually a combination of a few pictures, but these are some photos of once we actually got to our research transect. So there's a photo of Amanda outfitted in her safety gear. She's on board. She's about to do a net tow, which is something we'll talk about in a later slide as well. There's a photo of me doing some filtering of chlorophyll. And we'll get a little bit more into this in the future slides. All right, you can move to the next slide and I'll hand it over to Amanda.
Hello, everyone. So our day-to-day action kind of ranges depending on what type of science we're doing for the day. So these pictures describe an assorted array of tasks. So in the first picture, you can see that Kylie is collecting seawater from the CTD. And so that is something that would happen. CTDs typically go out early in the morning when the sea is nice and calm. And then that is the base of the water that we're working with for the day, because the pumps, when they go down, they take a lot longer. So the CTD is a pretty quick process where relative quick, an hour, 2 hours sometimes. But it has the basis of the seawater samples. Also, the next photo is collecting some biomass with a net. And so later on, we'll talk more about how big the net is and what we're capturing, but that is something that typically goes over the side. The center picture, which is where we are across from currently, is the Forward Dry Lab, and it is a kind of an information hub on the main deck of the ship, and it's showcasing photos of all areas of the boat as well as current sea conditions, weather conditions, etc. The fourth picture features Dr. Kristen Kimball and Dr. Isabel Dove, who are two other research fellows here on the Palmer. And so they are working on their incubation experiment there. And then as you can see on the far right, Kylie is reading the fluorescence of chlorophyll. Again, these are samples that are taken from the CTD, and one of the many tests is sampling the fluorescence with chlorophyll. So those are some of our day-to-day actions. Next slide.
All right, cool. So Amanda kind of gave a glimpse into what a typical day on board would look like for us in pictures. So here's a little bit more about the instrumentation that we were using to sample the stuff that we needed to collect from the sea. So this first picture on the left, this is a photo of the CTD. So that's our big instrument that we use to collect seawater. And this is a big instrument that goes down into the ocean, and we can control how deep it goes. And CTD stands for conductivity, temperature, and depth. So it's really a set of sensors that are on this big, we call it a rosette of bottles. And those are really just big water bottles that are there that you can see. They're just big water bottles, and they open at different depths as this CTD moves through the water column. And so we can collect water at different depths. And then whenever you can see in that second picture there, there's a bunch of us sitting around, and we're sitting on very high-tech 5-gallon buckets, and we're collecting water from these different bottles, because whenever we're collecting, we know which bottles came from which depths. And so then we can say, okay, I want, this much water from the closer to the surface, or I want this much water that was, about halfway down, or I want this much water which was deeper in the ocean. And so that was a big part of my job as we were really along that transect. And in this photo, the second photo here, someone remarked that it happened to be 5 women that were always collecting water from the CTD in the mornings. And someone remarked that we looked like milkmaids because we have these hoses and we're filling up our little containers, and we're sitting on buckets. So one day we really leaned into that milkmaid aesthetic, and I braided everyone's hair, and we all dressed in flannels and our muck boots, and we got some great photos. This middle picture here, this is a picture of one of our McLean pumps I think it's being brought up in this particular picture. But this is one of 4 pumps that it can go at different depths, but it goes deep into the ocean, and it pumps a bunch of water through it. And there's a filter that's inside that pump that then collects the biomass. So we're looking for, remember, we want to collect those diatoms that Becky mentioned earlier. So we're trying to collect as much of that biomass and as much of those diatoms and other things in the ocean that'll help tell us more about the questions we're trying to answer. In this fourth picture, this is a great photo of Amanda and another person we're working with on board named Tyler, and they have a giant net. It's kind of hard to tell in this picture, but Amanda is holding the circular part, that's the top of the net, and then Tyler is holding the bottom of the net, so it's really huge. And we take this and with the help of our marine technicians on board, we deploy this over the side, and that net collects everything that is flowing through it. And so the water goes through the net, but there's a little thing at the bottom that collects all the stuff that we want. So all of those organisms that we want to collect and study, they get trapped there at the bottom. So that last picture is then of some folks processing the biomass that has come through that net. So it kind of ends up being like a juicy goopy slurry of stuff, and it smells like a fish market because it's just a lot of goop from the ocean. But that goop is really essential to our operations and the science questions that we're trying to answer. All right, next slide. You want me to go? Or you? Yeah.
So, so once we get water and particles on board, we make a series of measurements, and we also are conducting some experiments. And so on the far left, you can see Kylie working on the filter setup, where we filter water to try to capture samples to measure sort of how much diatoms were there and how much chlorophyll was in the water column to give us an idea of the extent of primary production and of the sort of relative abundance of diatoms. In the next panel is Dr. Isabel Dove hunched over what we call carboys. So these are these big 10-gallon or 10-liter bottles. They have some stuff sticking out of them that allow us to put air and mix them up really well. And so these bottles are seawater that we took from one of the bottles on the CTD. And we give them a little extra food and light, and we grow them. So these bottles incubated for almost 3 weeks. So that we could make some measurements and do some tests on them when we get back on land. The fun thing about these bottles is that we grow them up and then we give them a little fluorescent dye so that we can see how they build their shells under a microscope later. In the middle is Dr. Kristen Kimball, and she is busy adding the fluorescent dye to small bottles of samples that she took either from the surface ocean or from some of those big bottles. And finally, on the far right is Amanda. And Amanda, so while Kylie told us that she measured chlorophyll throughout the trip, Amanda measured nitrate, so one of the key nutrients in seawater. And this is her pulling a sample through the nitrate analyzer, which she patiently did because it's kind of slow in comparison to chlorophyll. We spent a lot of time in front of that machine. Next slide.
So one of the things that was pretty fantastic about this experience is just learning how many people go into making this type of polar research happen. This is something that's made for every trip, but this is the crew that is on the boat currently with us. So you can see, we'll start down at the bottom. You can see the 10 of us that are represented for Becky's lab, the Robinson Cruise. Where you have Becky, Kylie, and I, and our 7 other researchers. And then moving up to the blue level is the Antarctic Support Contract. And these 10 individuals range from project coordinator, marine project coordinator, and marine lab coordinator, Layla and Chris, who make sure that we have everything that we need for our science, labs, equipment, scheduling, weather, communicating. Chris is our liaison between the boat crew and our science crew. And then amongst them are our marine technicians who facilitate getting the equipment on and off of the boat, which are incredibly important because they are very skilled mariners and they have a lot of experience working with science equipment on moving vessels, which is very, very helpful for us. And we are also assisted with our electrical technicians and information technicians. And they make sure that our sensors are working. They make sure that, for example, we have connectivity to talk to you all. And so they're critical in making sure that everything's working. Those big screens in the dry lab keep us informed. And so the Antarctic Support Contract group really facilitates our science. And then at the top is the crew of the Nathaniel B. Palmer, which is run through Edison Schwest offshore. And these individuals range from deck 2 engine crew, which means that half of them work on the bridge and work to make sure that ship operations go well, and the other half are in charge of the engine itself and the mechanics of the boat. So making sure that, you know, our engines and toilets flush, and we get fed, and the hulls stay clean, while also making sure that we are avoiding hazardous weather and making sure we stay on course. So without all of these groups of folks, this type of science cannot occur. And so it takes an entire team to make this type of research happen. And we've got a really great crew on the boat. Next slide.
All right, so we will kind of touch on a few of these things. There's a lot of information here. And we have some photos of said information in the coming slides, but we know people are curious about life on board the Nathaniel B. Palmer and what it's like to live on an icebreaker for the better part of 2 months. So we can talk just a little bit about like our accommodations. So we actually we all have a stateroom, which is a bedroom, and it has 2 bunks and a bathroom. And we were actually fortunate enough that we all have our own rooms on this particular cruise. So if we had a lot more people packed in to our ship, that might not be the case, but we actually were fortunate enough to have our own rooms. We get to do our laundry on site. So we all brought clothes, and then we were outfitted with some extreme cold weather gear that was issued to us. But we're here for a long time, so we have to do laundry on site. So we all do laundry every once in a while. That's all provided for us. I know a lot of our students and others as well will be curious about the food, and I've posted a little bit on my blog about the food, but we have a wonderful kitchen staff. So we eat in the galley that is on board the NBP, and we have people that are working throughout the day and night to provide us with 3 meals a day, so breakfast, lunch, and dinner, at kind of traditional times. And then whenever we're doing 24-hour operations, we also have a meal called mid-rats. So that's short for midnight rations. So Amanda, when she was working really hard doing those nitrate readings up until late in the night, got to enjoy those mid-rats as well. The galley is open all the time, and so we can go in even between meals. If we're hungry, we can get some snacks. They have a lot of the things that we would be accustomed to back in the United States, like popcorn and chips and cookies and drinks. But we also have been really fortunate to have what we call freshies. So that is fresh fruit and vegetables. We're kind of nearing the end of our freshies supply, but I saw Amanda eating an orange this morning, and it still smelled pretty good. So yeah, the galley has been wonderful. We have experienced some seasickness. Luckily, Becky informed us before we came of kind of her protocol for seasickness prevention and medication. So we all came prepared with promethazine as a common seasickness medication. And then there's also some patches that you can put on. We've all, I think, experienced different degrees of seasickness, but I think overall it's been pretty manageable, I would say, for the majority of us. We've seen some really cool animals, which I have some pictures of on the next slide, but I'll just tell you here, we've seen a lot of really cool birds. We obviously have seen penguins, so I do have pictures of those, but out here in the open ocean, we've seen lots of petrels and albatross. We've also seen some awesome mammals, so larger mammals that you might think of when you think of Antarctica, like we've seen some fur seals, we've seen some elephant seals, and they've been few and far between, but we have seen some whales. I've been fortunate enough to be part of the select group that have seen some whales. Yeah, do you want to talk about fun stuff and internet access?
Yeah, so when it comes to fun stuff, because we did have a long transect, we have several public areas on the boat So we can hang out and play games. They have disc golf and cornhole, as well as multiple different workshops that we have hosted. And as you can tell, the internet access, it's actually word on the street is this is the best internet they have had on the Nathaniel B. Palmer. And then
Starlink.
oh yeah,
Yeah.
and because we're using Starshield and Starlink to talk to you all, but we are all allotted a certain amount of data per day, and it cycles every 24 hours. But there are available desktop computers that have unlimited data for a specific login. So that is some of the stuff for onboard the MVP. Next slide.
And we'll try to turn our video back on once we get to the Q&A section. But for now, it's more important that you see the pictures anyways. We can all kind of jump in here. I put a few photos here of some of the awesome wildlife that we've seen. So those top the top two pictures there you can see are some penguin colonies. These were whenever we were in Palmer Station, so actually in Antarctica. We hiked over a glacier and we saw from a good distance, but we had some folks with really good cameras that captured some awesome pictures here of the penguin colony, and we could hear them squawking, and it was the wind kind of changed direction when we were standing there on the side of this glacier all of a sudden, and we could then smell the penguins. It's pretty smelly. But then the next day, a few of us hiked to a different location, and we saw some penguins kind of jumping through the water, and these are Gentoo penguins. So Gentoo, Gentoo penguins, they're pretty small, but they're very cute. Do you guys want to say anything more about these?
No, I think we're good. Oh,
So on the bottom,
sorry.
yeah, so on the bottom, these are some sightings of things that we can see at sea. And the Gentoo penguins that are jumping out of the water are in we're also at Palmer, but you can see this fur seal sitting on some pancake ice. I think that's fur seal. On some pancake ice that was flowing by us as we were somewhere in the middle of the ocean. And it's rumored, we don't know for sure, but it's rumored that we may have seen a very rare Ross seal with its pup floating by on some fast pancake ice that was moving quickly past us. We don't have great photographic evidence, but the pictures that we have suggest that it is a fairly rare sighting, which is exciting.
It's a really cool experience to be out on the bow of the ship or any part of the ship. But like Becky said, when we saw this rumored Ross seal with its pup, there was a few of us standing out there, and there's probably lots of wildlife that goes by us all the time, but we just happen to be out there at this one particular instance, and in this sea of white ice, you see this little dark spot, and we can pull out the binoculars and see this amazing animal that most people don't get to see in real life. All right, next slide. So we can all kind of jump in here too. Just a few select photos of things other than wildlife from our journey here. So this first photo in the upper left, this is from when a few of us were crazy enough to do a polar plunge at Palmer Station. We were not able to jump into the water, but we were given permission to run into the water off of the boat ramp, and it was snowing. And that was the worst brain freeze I've ever had in my entire life. And then we had to run through the snow, and then we immediately all went and got in a sauna. And I guess I'll talk about the picture below it as well. This was a picture taken as our group was hiking over that glacier when we were in Antarctica at Palmer Station. I don't know if you guys want to talk about other photos.
The center photo with the sunrise, that was taken approximately 2:30 in the morning, several weeks back. Right before it, we saw the rare green flash occurrence, which is pretty amazing. Down below is celebrating Halloween. You can see assorted costumes were featured, and that is the Robinson Science Crew. And then to the right are a couple of variations of icebergs that we see as we're floating in the ocean. Top one close to Palmer Station, and lower right, something that we passed, an iceberg we passed last week as we were coming back north. Next slide. Oh, go ahead.
Yeah, so one of the sort of parts of this project isn't pure science. It is a collaboration with one of our science team members, Georgia Rhodes, from the Rhode Island School of Design. And it's an exploration of arts-based visualization of scientific research. And so Georgia came on board with us and ran a 4-part workshop to engage the science team as well as some of the Antarctic support contractors in art. And we did some art immersion, and we had to think about and talk about and do art on the ship. And out of that 4-part workshop in which we explored different ways of sort of thinking about how our science and art overlap, we came up with some proposals, and shipboard participants who are interested will be paired with Rhode Island School of Design art students to collaborate on artwork that will be exhibited in the fall of 2025. And so this was part of our original National Science Foundation grant. And it's been unbelievably successful so far, and met with a lot of enthusiasm from both the science team and the Polar Steam Fellows, as well as the marine technicians. Next slide.
So as we think about, we're coming to the end of our time on the NBP here. We are, so technically we are a day ahead of you all. It's December 3rd for you all, but it is actually December 4th for us, which is kind of weird to think about. We are scheduled to get into our port in Littleton, New Zealand on the 6th. So here in a couple days, we'll be reaching our port. And once we do that, we're not just getting off the ship immediately. We have to kind of deconstruct our labs that we've set up so that the ship is ready to go for the next science cruise, which will depart just a few days after we take all of our stuff down. So we'll get into port, take our stuff down, and then that's not the end of the story for really anybody. We've collected a lot of samples here at sea, and those samples will get shipped back to URI, so University of Rhode Island, and we also have collaborators at the University of California, Santa Barbara. So people will be getting samples, and the science will continue to happen in labs with the stuff we've collected here. We will also be continuing, so Amanda and I will continue to collaborate with Becky and with the other folks that we've worked with as we create our educational resources that we will be making for Polar Steam. And then as Becky just discussed, we'll be continuing to collaborate with this art and science kind of merging of these two things to make our work that'll be exhibited in the fall of 2025 for Visathon. Next slide.
All right, so
Q&A.
that is the end of our presentation. Thank you all for your patience. I'm going to turn it back over to Polar Steam and go with the Q&A.
And we might try to turn our video on, and if it doesn't work, we might have to turn it back off.
Excellent. Thank you so much. That was great. Really loved learning about the science that you guys are doing aboard the ship and a little bit about life on the ship. So we've got a few questions for you. We'll jump right into those. So we've got actually, Kylie, I think one of your students, Elizabeth, who wanted to know about sleeping on the ship and if you ever wake up from like the waves and the water at night. And then if you also sometimes get like cramped and crowded on being on the ship the whole time.
Yeah, great question. I think this is my student age, so thanks for submitting that question, Liz. Yeah, sleeping. So it is like a twin size bed. And I could have chosen either top or bottom bunk. I chose bottom bunk. And I actually sleep really well on the ship. There are definitely times where the waves are so much that, my body is physically moving back and forth, and that makes it kind of difficult to sleep. But actually the gentle rocking of the ship.
Oh, I think we're freezing again. We may need to do camera off.
Crowded on the ship. I'm trying to think if there have been instances where I felt crowded. We did have more people on the first few days of our journey before we dropped them off at Palmer Station. But even with those folks on board, the ship is able to accommodate a large number of people. So I have not felt boxed in.
And you can always go outside and the view is endless. So there is well, you can't always, but
If the weather's good.
when the weather is good, you go outside and take in a view or go to the bridge and stand over to the side and nobody will talk to you. You get a little space.
Excellent, excellent. All right, so we have a class, Mrs. Ramey's class, fifth grade, and they are studying plants and photosynthesis, and they know that the plants are releasing oxygen through the stomata. So they're wondering how diatoms release oxygen back into the environment.
Ooh, that's a really good question, Mrs. Ramey's class. So I might turn this over to Becky, who
Uh-oh, we're freezing again. Yeah, thank you. Okay. Can't hear you guys yet.
The diatoms live submerged. They don't actually have stomates. So instead they have gases that diffuse across their cell membrane. And so it's just more free flowing, I guess you could say. They definitely are photosynthesizing, but in a different sort of cell structure.
yeah, cool.
Perfect, thank you. And then they also want to know what the RIS level is for what RIS level diatoms have.
Gonna try to turn the video back on for this one. I think that diatoms have a RIS level. If we're talking like on a scale of 1 to 10, I'm gonna say like a strong 9 out of 10, because RIS means that you are charming and that you're good looking. So diatoms are very charming. They come in so many different varieties and they're so cool to look at under a microscope. We showed you a few pictures of microscope pictures.
All right, so they're talking about microscope pictures that we saw on the slides. See if we get them back here in a minute.
We can hear you.
Okay, I can hear you again.
High Riz. That's the summary.
Yeah, did you
Perfect.
get my Riz level answer?
Yeah, we got 10, and so we got real high res. Yes.
Okay.
Okay. We're wondering, we've got another question about what surprised you about Antarctica?
Freezing.
Oh, can you guys hear me? All right. Let's see. Looks like we're having, looks like we're having some issues with the internet on the ship. So, oh, they might be back here, but we're in the middle of the Southern Ocean, so I think this is to be
Are you there?
expected. Yes, we're here. Can you hear me? Right. Well, we've learned a lot today. I don't know if we're gonna be able to do any more Q&A here. Let's see, we'll give them just another minute and see if we can get them back. Oh, okay. I think they're trying one more time to come back in. We'll see if we can get them. But
Can you hear us?
yes, we can hear you.
Okay.
Okay, so they want to know what surprised you about Antarctica.
For everybody else.
All right. Well, we may need to wrap it up here. So I'm going to go ahead and do that. I'm going to share the slides again. And
All right, we can see what you're doing now. I think we must be in a slightly less internet access zone here, but
okay. Do you want to try one more question?
oh yeah. Let's go.
Okay. So they want to know what surprised you about Antarctica, and is it what you anticipated?
I'm gonna answer.
Ooh. What surprised us about Antarctica and what was the last part?
And is it what we needed?
Is it what we anticipated? Well, this is Amanda, just in case you forgot what my voice sounds like. I've never been south of the equator, and so I had no idea what to expect. And starting this journey in Punta Arenas, Chile, was just eye-opening. And so when you're crossing the Drake Passage, you don't know your left from your right with the seas, but then all of a sudden you start to see icebergs and you're like, is this real? Oh my gosh, that's an iceberg. And then we pulled up to Palmer Station staring at a glacier and setting foot on the seventh continent of the world, and it just blew my mind. It is more than I could have ever thought it was. And as much as I want to recommend everyone going there, like don't go there. Also, just because it's such a vital resource for our entire globe. And I think that if you have the opportunity to go and see it for science and to see how important it is for us as a global population, I think just knowing how massive it plays a role in our lives is enough to set an experience in your soul forever, core memory. So 100%. And I still forgot the second part of the question. So
Yeah, like was it what we hated?
for me, it was definitely more than I anticipated. I did not anticipate getting to set foot on land in the initial part of this fellowship. And so the fact that we got to stand on Antarctica and sail along it and see all of the ice was pretty fantastic. So I am super grateful for this opportunity.
And I'll chime in with a couple
Actually,
of things.
Kylie, I'm gonna reframe the question just a little bit for you.
Yeah.
So there's a North Bend, Oregon class, fourth grade class that wants to know what inspired you to participate. So I think it's a similar question.
Yeah, for sure. Well, I think it's different answers for Amanda and I both. But for me, I live in the middle of the country. I live in Kansas and my students do as well. And the nearest coastline to us, so the closest place for us to get to the ocean is about 18 hours away. It's about 18 hours for us to get to the Gulf of Mexico and even farther to get to the Atlantic or Pacific Oceans. So a lot of my students have never seen the ocean and, may not get the opportunity to for a long time. But there's really important things that the ocean controls for all of us. There's a lot of matter cycling and there's a lot of climate patterns that are really important to the whole world and even us in Kansas. And it's kind of hard to visualize those things, or it's hard to see the relevance or the impact that those things have on us being so far away. So what inspired me to participate is bringing that important science that's happening to life for my students. I get to be a kind of a vehicle to make it more real.
Awesome. Thank you so much, Kylie. I think this one is for Becky, maybe. Can you share something interesting about polar diatoms and how they might differ from tropical ones?
Well, so diatoms are excellent environmental indicators. And so the diatoms that we see down here maybe are assemblage, one of our graphs kind of alluded to this. They're very particular about where they live. And so they're tuned in to the temperature or the salinity, how much salt is in the water. Some of them really like stable water. They like to sit up in that sunlit surface. And some of them don't mind being mixed around. And so the polar diatoms really close to the coast are usually sea ice diatoms, and they are these tiny little they're these tiny little microscopic organisms on the order of, say, 10 microns, so a 10,000th of a millimeter. Is that right? I think I got that right. So tinier than a grain of sand. And they thrive in the sea ice, and some of them are actually trapped in the sea ice, and then they come back out and they seed the water column after the ice melts. And then as you move further north, you get diatoms that are bigger, and they are adapted to this really high nutrient, high silica environment. And we see some of those same organisms as we get towards the tropics. The species differ, but only in places where there's lots and lots of nutrients. So off the coast of Oregon, for example, there's an upwelling zone, and we can see some of the related diatoms that like these high nutrient conditions. But most of the tropical diatoms are, the ones I think of from the open ocean are ones that have adapted to lower nutrient environments. Some of them can move up and down in the water column. So they have they don't rely on the environment to keep them in the sun. They actually can go down and get nutrients and then come up and get sunshine. And so they're generally bigger in the
Awesome.
tropics.
Thank you. Let's see, just a reminder for folks to post questions in the chat if you have them. And then let's see, we're wondering, what is a good technique for counting diatoms on a moving ship? Is that hard?
Take lots and lots of breaks is what we've been told. So we've all looked under the microscope on the ship, but one of our science team members, Isabel, is a master. And she is our taxonomist, meaning she can identify all of the different species and she counts, I don't know what the count is now, but it's thousands of diatoms on the microscope. And so you have to take lots of breaks and not move your eyes between, like, say, the computer and the what you're looking down the microscope. You have to like focus on one thing, I think, is the key technique. She does fix her slides so they're not moving around, but sometimes we look at things just in a drop of water, and that adds another element of sort of stomach turning activity to the whole thing.
Lots of patience. It's required.
Nice. Will you also take some samples home from the cruise?
Yeah, so one of our key tasks in the next few days is to pack up all of our samples. And so we've got hundreds of water samples, samples of particles from the pumps of different kinds. We kind of split them up into various bottles for different analyses when we get back home. And those net-to samples will be used for experiments. The samples that Kristen put the fluorescent dye into will be brought home, and we'll make slides and look at them under a fancy microscope. So we have lots and lots of slides that we'll be able to continue counting diatoms on land. I have a feeling it will go much faster on land.
And it's kind of an ordeal to get these samples back to the United States. We were just overhearing a conversation with one of our team members who is taking things back to be to be sent off for DNA and RNA analysis, and those samples have to be cleared by the CDC to get back. So there's a lot of moving parts into getting these like biological samples back to the US.
Wow. Wow. All right. We have a couple of questions about like bringing things back to your classroom for Kylie and Amanda. Can you think of one new thing that you maybe haven't done before, but that you'll bring back to your classroom?
Well, one of the really cool things that we picked up from Georgia Rhodes is we had a really fantastic zine workshop. And for me, I made a really nice informational zine about our crew, you know, like I explained in that one slide. So it breaks down information about the Palmer boat itself, the ECO crew, the ASC crew, and the science crew, as well as a little bit of history about things. And I would really love for my students to be able to break down information and create a really simple but informative handout, you know, because this is, you know, trying to explain concepts of science or information about this cruise. There's so much that goes into it, but you know, bringing back the experience of taking information and making it into smaller digestible amounts with cool pictures and kind of a fun flair. That is definitely something I am looking forward to bringing back and testing in my classroom. Informational fun zines.
It's a great way to demonstrate understanding. It's something different than a poster or a slideshow or a presentation. Zines are something we really, really enjoyed making and are excited to share.
That's so cool. Maybe could you just explain for people who don't know what a zine is, like, how would you say it in like one sentence?
It's like a self-published homemade magazine. Generally it's crafted from you start out at least with one sheet of paper that's folded and cut in such a way that you can make your magazine, and then you can unfold it and photocopy it and then bind it up and distribute it.
Cool. Thank you. Kylie, did you want to add anything else? Or should we go to another one?
Next question.
Okay, perfect. We have some folks who want to know what your favorite part has been, favorite part about Antarctica or being in the Arctic Ocean. Southern Ocean.
It's really hard to choose. We're all like looking at each other right now for some ideas. I think if I had to choose one thing, while the scenery is incredible, I think my favorite thing has been getting to work with people who have such vast amounts of knowledge that is different than mine and can teach me things that I don't know and can teach me things that I can take back to the classroom and share with my community.
That's great. Thank you, Kylie. Amanda, any thoughts there?
I think, like I stated before, I think my favorite part is just having the opportunity to see a place so untouched that has such a huge impact on our entire globe. And just really seeing a super zoomed out lens of just our global dynamics with climate and ocean currents and oxygen production and carbon sinking, like just really all of these things that you learn in science when my students say, you know, how am I going to use this when I grow up? You know, these are important concepts and ideas, and being able to be here and witness it firsthand, hopefully will be such a huge testament to my students and all students that this stuff does happen beyond a textbook or a lesson in a classroom. And I think having lived this experience and coming back and being an artifact that I can present myself to my students, I think that's been my favorite part. It's just living and breathing what we have been learning about since, you know, I was in school. So I think that's been my favorite part.
Nice. All right, so we're gonna wrap it up here. We'll just have you guys send us out with one more sort of highlight. So maybe for each of you, if you could pick a highlight, you know, something that happened on this cruise that you're really jazzed about, excited about, that you can share with us.
So this is Becky, and I'll jump in with a science highlight. And so this isn't the first time that I've come down to this particular location. And so for me, we were sort of in the heart of the Southern Ocean along our transect right during the bloom. And so even though I predicted that it would be here, it's so nice to like actually find what you're looking for. And it was just really exciting to know that we were here getting the material that we wanted to recover.
That's fantastic. Nice.
I think one of the highlights for me has been getting to work with so many amazing people, namely women, that are really at the forefront of their fields and making science and inquiry possible for women everywhere. It's been we have men here too, but our chief scientist is a woman. Many of our PhDs that we're working with are women, and they have been really patient and inviting to, you know, teach Amanda and I the things that we need to know to be on board with and up to speed with the science that we're doing, and they've been really inspiring for me.
Nice. Thanks, Kylie. Yeah, Amanda.
A highlight for me, going back to all of the people who make this type of science happen, one of my biggest communications to my students is you don't have to go to college to work in STEM. You know, it's not required to go to a four-year university, even though I do live in a college town. But there are people on this boat who went to maritime school, who have trade school backgrounds, who are integral to making these ship operations happen, be it working on the machines, working on the computers, making sure the boat runs. And so seeing the diversity of backgrounds from all of the individuals who work on this boat and allow this science to happen has definitely been a highlight, and just really hammering home to me what I'm telling my students is so true in all of these jobs.
Nice. That's awesome. Thank you, Amanda. Before we let them go, I'm just gonna mention that you all can follow along. Here are some of the links if you want to follow Kylie's blog, Amanda on Instagram, and Southern Science in the Southern Ocean. Is that another collaborator on the ship?
Yes,
Becky's lab?
yes, that's URI's GSO lab.
Perfect. And then real quickly too, there's lots of ways to follow along with Polar Steam. So please check us out. Our link tree has all the links to all the things that you want to follow along, including our social media and our blog. So I'm going to stop sharing, and then maybe we can have you guys turn on your camera and just say goodbye. All right. Thank you so much for today. We really appreciate it. Thanks for everybody hanging in.
Of course.
Awesome. Okay, I think we're done. All set. Thanks so much, everybody,
still
for
here.
joining us. Oh, you're still there.
Oh,
Okay.
looking at it.
Bye-bye.